Here we are in the first lecture of Unit 7, called Make It Your Own. The quote from Rosabeth Moss Cantor: "When you fail at something, the best thing to do is think back to your successes and try to replicate whatever you did to make them happen." Our learning goals are to conceptually describe DNA replication, detail the process that is DNA replication, as well as explain the importance of the components. What we already know from previous units is that information is in the sequence itself, that nucleotides encode the info inside of the nucleic acids, and that base pairing rules, the A to T and the C to G, as well as enzymes, are what allow information to be copied. We learned about that when we looked at protein synthesis and how the processes of transcription and translation worked. In the lab, we really tackled an age-old philosophical problem that's very relevant to what we're doing here. We learned about the complexity that is identity. Wherever we explored the ship of Theseus, we learned that, old and new is hard to pinpoint as this or that, and there's always shades of gray in between, and you can be both at the same time. And that parts can be replaced, but still maintain that identity. So, in DNA replication, we know that the genetic information is stored as DNA. And we know that we need to continually produce more DNA as an organism changes and grows. I mean, just look from a small child to a grown adult. The extra cells, they're going to acquire more DNA themselves. And all these cells that keep getting made, the DNA codes for all the proteins, and the proteins are what do everything. If a cell didn't have DNA, it wouldn't be a cell. It would die very quickly. So where does all the DNA come from? Well, we need to keep making more sets of DNA. Whenever it's time for a cell to grow, for it to make a new cell, for it to replicate, the DNA must be replicated, because the cell needs a set for itself, and will need to give a new set to the new cell. So, we've talked about the idea of base pairing before, and we talked about biomolecules. This here is the original paper as it was published in a scientific journal, very famous, with the very generic name Nature. You can see the publication date is April 1st, 1953. This is almost the entirety of the paper. There's only another paragraph on the other page, and then some citations and signatures and that. It's almost all right there. Very famous drawing right there. This is where Watson and Crick note the structure itself for nucleic acids. And it's important to note that, we identified this as a chemical structure. We didn't have to personify DNA that it wants to do something or that it chooses to do something. Those bases pair because of largely a geometry issue. The way that the chemical bonds form between them, A and T have two spots to bond. We can see there's these two hydrogens that are hydrogen bonding with these nitrogens. And then in G and C, we see that there's three spots. So they fit together nicely. One of the last sentences in one of the last paragraphs, the paragraph is a single sentence itself. Watson and Crick wrote this. That's a picture of them there at a lecture where they're talking about their double helix shape. Look at this sentence. "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." That's powerful. What do you think they're suggesting? Well, we've learned a lot since 1953, so let's talk about the process. So, first of all, the DNA that's getting copied, that this pairing rules is letting happen is happening by proteins that do it. The DNA is getting copied, but it's passive in it. It doesn't actually do anything. The structure of the DNA, that organization, the fact that there are rules, the fact that A only fits with T and C only fits with G, that's convenient. And what that lets us do is let the proteins be able to perform their functions. In this case, we have proteins such as DNA helicase. That's the very first protein that we're going to talk about. Just based on that name, what do you think DNA helicase is? What do you think it does? Well, the fact that the name ends in ACE lets us know it's an enzyme. We like to name enzymes in ACE. It lets us know that they do something, that they facilitate some kind of a reaction. The fact that it's called DNA helicase lets us know it has something to do with the helix. In this case, what's happening is we can kind of make an analogy to a zipper. It's unzipping, meaning that it's breaking those hydrogen bonds that connects the two bases. So it's separating a double-stranded molecule and opening it up so we can get to the insides. Now here's where that copying mechanism that Watson and Crick said was immediately suggested by their suggested pairing is that we have a new enzyme. This one's called DNA polymerase. Note that ACE there on the end. We see these two words, poly and mer. Poly means many, and mer means part. So what this enzyme is doing is dealing with many parts. In this case, putting them together. So our DNA is unzipped. We have two exposed sides. Now, there are extra free-floating A, T, C's, and G's, adenosines, thymines, cytosines, guanines. They're just floating around, not attached to anything. So what DNA polymerase does is attaches one of those free nucleotides to the open side of the strand. So we can see in this diagram, this is a simplification. You know, it's a brief diagram for ease of looking at. So the helicase, we can see there on the left, is separating the strands, opening them up. And then we can see on the right, the DNA polymerase is tailing behind, catching up with it. And what it's doing is taking those free nucleotides and just matching them with what's there. If there's an A, it grabs a T and puts it on. If there's a C, it grabs a G and put it on. And it's slowly filling it in. Now, if you remember, DNA has two sides. The polymerase has to work from the 5' to the 3' side. It's the only way that it'll work. Now, this leading strand is easy because the helicase moves forward and the polymerase moves forward. They're both moving forward in the same direction. Now, the problem has the other side because there's two sides that have been opened up because it's a double-stranded molecule. So if helicase unwinds it, we have one side that needs to be filled in, and that one's filled in easy as the polymerase just goes 5' to 3'. Well, on the other side, you can't go backwards 3 to 5. So what you have to do is you make a few pieces going the wrong way, and then you jump back, make a few pieces, jump back, make a few pieces, until you're slowly catching up again. So that's the leading strand and the lagging strand. The leading strand is really easy. In this case, in these pictures, the overall direction of replication is moving to the left, because right there at that fork is where helicase is working. On the top one, in this drawing, that's the leading strand, meaning that DNA polymerase is moving in the same direction that DNA helicase is moving. The bottom strand is the lagging strand, meaning that polymerase has to move in the opposite direction. So it moves to the right until it finds the end, and then it jumps back to where Helicase is gone, catches up with Helicase, and it goes backwards and fills in all the stuff it skipped, fills it all the way back till it gets to where it started, where it was at before, jumps ahead again, back and forth, back and forth, back and forth. It makes it in pieces. These pieces are called Okazaki fragments. Another enzyme called DNA ligase goes back forward and makes sure all those little fragments get joined together and smoothed out into one solid strand. So the leading strand, the one that's going in the same direction of Helicase, 5' to 3', it sets the pace. It's going along, just chugging along. The one that has to go backwards, it's 3' to 5'. So it's the one that's really struggling here. So it falls behind and it sprints forward to catch back up, but then it has to go back and fill in the blanks, and it sprints forward and back and forth and back and forth. That's where the trouble is. So, developing question, A1. Why do we only see DNA ligase on the lagging strand? Why does it not go over the leading strand? Well, the leading strand is already one continuous piece. It doesn't have a need for DNA ligase to go over and fix it up, because DNA polymerase on the leading strand is moving in the same direction as DNA helicase. So it never has to detach, it never has to backtrack, it never has to play catch up. All right, our proficient question. If you copy down someone's phone number, you might make a mistake if you're going too fast. Well, DNA replication happens fast. Why doesn't it make a bunch of mistakes? So this is what Watson and Crick was alluding to, and they said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Here's the thing they're talking about. A goes with T. C goes with G. A doesn't fit with G because it has the wrong number of hydrogen bonds. So there's an inherent mechanism that chemically makes sure that they connect in the right way. It's just a geometry. It's a, square peg in a round hole problem. A master question. Speculate on a reason why DNA polymerase needs to work in the 5' to 3' direction. Why is it so needy? Well, the enzyme activates at the 5'. That's where it binds, and that's how it wraps around. It's a shape thing. It's another geometry problem. Remember that we learned about proteins is that they're formed from the way that the amino acids wrap up and form a very specific shape so that they bond in the right way. The shape of the DNA matches the shape of the enzyme. You can't fit it on backwards. Now here's something that's interesting. You start with one DNA. You go through replication. You end with 2 DNA. But there isn't a new DNA and an old DNA. How is that possible? Well, the vocabulary word we use for this is semi-conservative. So think about how DNA helicase unwraps the double strand. That means we have the leading strand and the lagging strand. Both of them contain 1/2 of the original strand. Both of them only have 1/2 that was rebuilt. So you can think of whenever we end, we have 2 DNA molecules. Each of them has one side of the backbone that's old and one side of the backbone that's new. So from my original DNA to my new DNA molecule, it was semi-conserved, meaning we kept part of it, but not all of it. Now, DNA polymerase makes few errors because of the base pairing rules, but it does make a few. It does make them sometimes. Sometimes it gets wedged in there and it might not fit quite right, and it happens. They're rare. But the enzyme also does proofreading. So not only is it very unlikely to happen at the beginning, but it even goes and checks, and if there's a problem, it even goes and fixes it. Proofreading is what that's called. Now, I know it's called a mistake, and mistakes sound bad, but mistakes are important. They cause change. Changes can be good and changes can be bad, but changes keep things interesting. In 1 billion nucleotides, DNA polymerase will make one mistake on average. So when do we see DNA replication? All the time. Constantly. You are currently, as you are listening to this lecture, you have cells going through DNA replication. We are in a constant state of renewal. Now some cells are going through it and some cells aren't. We'll talk about the stages of when cells are doing what, but all the time some of your cells are doing this. It's mind-blowing to think about, the constant state of renewal that we're in. We're like rivers. So, a developing question. What would happen if DNA polymerase was graded on its assignments in class and it made an A, but not an A+ on its DNA replication assignment? Well, let's think back to what I said earlier. DNA polymerase, on average, can do a billion problems in a row, a billion pairings, and only make one mistake. Think about it in terms of your class. If you did 100 problems in a row and only made one mistake, you'd get a 99%. We consider that to be an exceptional score. Think how much better DNA polymerase is. 1 in a billion. You and I are proud about 1 in 100 mistakes. If we only do 1 in 100 mistakes, exceptional. Think if DNA polymerase made 1 in 100 mistakes. The DNA would be so different, we would get huge, dramatic changes that probably wouldn't work out very well for the proteins whenever we needed them. Here's a proficient question. Human DNA has approximately 3,234,830,000 base pairs. Now, approximately the rate at which DNA polymerase can replicate is about 50 base pairs per second. How long would it take for DNA polymerase to copy a single set of DNA? Well, if we take that 3,234,830,000 number, divide it by 50 base pairs, that'll get us 64,696,600 seconds. Well, almost 65 million seconds if there's 60 minutes in a second, and 60 seconds in a minute, 60 minutes in an hour, 24 hours in a day, 365 days in a year. Well, that comes out to just over 2 years that it would take for DNA polymerase to make just one copy of DNA. Now, in reality, a cell's complete set of DNA can be replicated in a few hours. If the previous slide gave quality approximations, and they are, we looked them up in real research, how can this be true? Well, we made an assumption. We made an assumption that only one DNA polymerase would do the job. And that's not how it goes. Lots of DNA polymerases work all at the same time, just at different spots. So the picture to the right is made using a really cool piece of equipment called a tunneling electron microscope. Expensive, powerful, awesome. Each of those bubbles where you see the arrows at, that spots where the DNA has been opened. Those are sites of DNA replication. So in this image, that thick strand is a strand of DNA, and there are 3 DNA replication sites happening right here, where DNA helicase has opened it up and polymerases are working. So one polymerase on its own might take 2 years, but a bunch of polymerases together can do the whole thing in a few hours. Now, what's interesting is that all life on Earth replicates nucleic acids using nearly the identical method. That we've seen all the parts, we've seen the whole process on how DNA gets replicated. There's a lot of parts in that, a lot of steps in that whole DNA replication thing. What is this evidence of? Well, it's evidence of common ancestry. The fact that this information and these chemicals had an effective way to copy themselves allowed them to spread. That this molecule, the pairing rules that were suggested by Watson and Crick, allowed it to be made over and over again with a good degree of precision. And since they encoded for proteins that, one, some of those proteins are the polymerases that copied itself, and 2, were traits that did other things that gave it an advantage, this whole process kept getting passed on. It was passed to each cell thereafter. And we haven't had any changes. No organism has found another way to do this process. There's never been an advantage change. All things on Earth have a common ancestry with an ancient cell that developed this methodology by random. And the first versions were crude, and DNA polymerase has improved in its accuracy and its speed, and the other enzymes and the other things have been refined. It wasn't a nothing to everything. There were small steps and small changes that have added up over time. But this is one of the many, 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 many commonalities that life on Earth shares due to common ancestry.